A quick review: the pharmacology of agents used in the treatment of asthma. In this slide, I'm going to summarize the pathophysiological changes that will be taking place in the classic allergic asthma. So first of all, we have the exposure to allergens, which they are foreign proteins coming from different sources. It will be taken by the immature dendritic cells, or what we call it, the antigen capturing cells. Then it's going to process the antigen and will be converted to a mature dendritic cells. Those mature dendritic cells now, they are going to be the antigen presenting cells. They are going to present the antigen through a specific placement in the major histocompatibility complex class 2. And that one will be now binding to a naive CD4 T cells, which containing in them a T cell receptors. Now, these T cells now will be inducing the T cell differentiation into the Th2 subtype, the helper cells, type 2. And the helper cells type 2, it will be producing two actions. One of them, it will be releasing the interleukin 5, 9, and 13. They will be producing it in the airway epithelium and also in subepithelial mucosa. And that will be leading to accumulation of specific white blood cell, eosinophils, and basophils, and the mast cells. The other function, it will be to induce the goblet cell metaplasia and also the mucus production. Now, the helper cells type 2, the Th2, is going to generate cytokines which will be promoting the B cells to produce the immunoglobulin E. And the B cells, another name for it, it is a B lymphocytes. Now, these cells now, it will be converted to plasma cells, or stimulating the production of plasma cells, which will be producing the immunoglobulin E. And the immunoglobulin E is going now to be attached to a specific receptors. Those the one we call it the Fc epsilon R1. This receptor, I'm going to explain it in a little bit more details in the coming slides. So now the immunoglobulin E will be binding to these receptors. So now whenever we have an antigen binding to the immunoglobulin E, is going to activate these submucosal mast cells and will be releasing different types of substances like histamine, cytokines, tryptase, leukotrienes, C4 and D4, and prostaglandin D2. Now, these substances will be leading to a contraction in the smooth muscle in the bronchi, and also will be leading to a leakage in the vascular system. Now, in this slide and the following slide, I'm going in a little bit more detail. So we have the immature dendritic cells, or the antigen capturing cells. Whenever they are interact with the antigen, they are going to engulf it. So the antigen will be inside them, and that will be either by endocytosis or phagocytosis or different mechanism. After that, they are going now to be changed to the mature dendritic cells. And here we have a dramatic change in their morphology, a reduced uptake of the antigens, and also impaired the antigen processing activity. And also the mature dendritic cells now will be exhibiting a strong co-stimulatory and T cell activating capacity. Now, if we look about the mature dendritic cells, now after they are interacting with the antigen, if we look about their cell membrane, we will find that they are going to now produce multiple pieces of the related to the antigen. And that is going to be, here we have the cell membrane, and they are going now to present the piece of the antigen inside a major histocompatibility complex class 2. And here I'm going to draw it, the major histocompatibility complex class 2, MHC2. So it is composed of two subunits. They are spanning the cell membrane. We have the beta and the alpha subunits. Here we have the disulfide bond, as you can see it. And here we have in the beta 1 and the alpha 1, they are the site where we have the peptide binding cleft. So here we have the peptide that is going to had been ingested, and it will be now presented to the other cells. So this domain for the beta, we said that we have a beta 1, beta 2, and alpha 1, alpha 2. So now again, for the mature dendritic cells in their membrane, we found that we have the major histocompatibility complex class 2. Again, I'm going to draw it again. So we have two molecules that will be spanning the cell membrane. We have the beta and the alpha. And again, here we have the disulfide bond. And we have the beta 1 and the alpha 1 where we do have the binding site for the peptide. So here we have the peptide already bound there. And then we have the naive now CD4 T cells. These cells, they have in their cell membranes, they have specific receptors. We call it TCR, T cell receptors, that whenever these receptors now will be interacting with the peptide, that is going now to induce the T cell will be activating these naive CD4 T cells, and those will be whenever they activated, they are going to induce the T cells differentiation into two types of helper T cells, 1 and 2. So here we are dealing with the type 2. So here we have the type 2 helper T cells. They are going to generate different types of substances, cytokines. These cytokines, they are actually a specific protein, will be secreted by these helper T cells, and they are actually a signal molecules. So here they are going to produce, for an example, different types of interleukin. The interleukin, we have around now about like 36 different types of these proteins. So here, the major ones that will be producing an effect, we have interleukin 5, 10, and 13. So those, they are going now to promote the B cells to produce the immunoglobulin E. And the other name for the B cells, it is a B lymphocytes. So that will be now promoting it to produce a plasma cells, which will be responsible about the production of a specific immunoglobulin E. And here I'm going to draw the structure of the immunoglobulin E, composed of two chains. One, this is a green one, we call it the heavy one, and the red one, we call it the light chains. And here we have the disulfide bonds joining the two chains together. And here we have in the red one, in the light chains, we have the top area, it is the top domain, it is a variable, and the other area, it is a constant. The same for the green one. We have the top one, we have a one variable region, and we have a constant region 3, 1. We have 3, number 2, and 3, number 1, 2, and 3. These are a constant regions or constant domains in that immunoglobulin E. Now, in the top of these domains here, we have the binding sites for the antigen. So here we have the cell membrane for the submucosal mast cells and the basophils. And here we have the receptors on them. That's the one we call it the Fc epsilon R1. Now, these receptors, they are high affinity receptors for the Fc region of the immunoglobulin E. And I'm going to show you what is the Fc region of the immunoglobulin E. So here we have one receptors and another receptors located beside it. Then we have the interaction with the immunoglobulin E. So here we have an immunoglobulin E, and here we have the Fc region of the immunoglobulin E, which it is what we call it the fragment crystallizable. So here we have two immunoglobulin E already bound to two receptors, and we have in between them we have the binding for the antigen. So whenever antigen will be binding to two immunoglobulin E, is going now to induce changes and will be activating these mast cells and will be releasing different types of substances. So they will be releasing histamine, they will be releasing inflammatory cytokines. Again, cytokines, they are proteins that they are acting as a chemical messenger. And also they will be releasing the tryptase, which it is a serine proteinase. It is an enzyme that will be cutting the peptide bond in proteins. And also they are going to release the leukotrienes C4 and the one D4, also leukotriene D4. Besides, they are going to release the prostaglandin D2. And all these substances will be producing the following effect in the next slide. So now let's make a quick look about the airway in asthma. So we have the smooth muscle hyperplasia and hypertrophy. We have swollen epithelium. We have a decrease the diameter of the lumen. We have an increased amount of the mucus. Beside that, we have a hyperplasia in the goblet cells. And we have a subepithelial fibrosis. So we have a deposition of the collagen. Beside that, we will have an increase in the volume of the submucosal glands. and also we have an increased number of blood vessels. This just like to give you a general idea. So we already described the classic allergic asthma from the exposure to the allergens until we reach to the point that we are going to release different chemical mediators like histamine, leukotrienes, prostaglandin, cytokines, and so on. Now, the release of these chemical substances will be leading to a contraction in the smooth muscle in the bronchial tree, and also will be leading to a vascular leakage. Now, these two conditions, the leakage through the blood vessels and the contraction of the smooth muscles, will be actually responsible about what we call it the acute bronchoconstriction, and that will be happening in the first few minutes up to about like 60 minutes. So that's what we call it the early asthmatic response. Then we have the helper T cells, as we have seen, that they are going to produce different types of interleukin 5, 9, or 10, and 11, 13. And that will be this interleukin will be produced in the airway epithelium and in the subepithelial mucosa. And that's, as we have seen, is going to accumulate the different types of white blood cells and the mast cells. Beside is going to induce the goblet cell metaplasia and increase the mucus production. Now, these two incidents will be leading to what we call it the more sustained phase of the bronchoconstriction, and that will be what we call it actually the late asthmatic response. Usually will be taking place after about like 3 to 6 hours of the asthmatic attack. And now let's say a few words about the bronchial hyperactivity, or what we call it the bronchial hyperresponsiveness, which will be defined as an excessive bronchial narrowing. So we have a severe bronchospasm that will be manifest as an exaggerated bronchoconstriction response to the airway of different stimulus, like for an example, a viral infection, exercise, cold air, cigarette smoke, or activation of the neural pathways. And this bronchial hyperactivity considered to be a hallmark of the inflammation in asthma. And actually, it will be related to the severity of the disease.